present, and thank you very much.
Great, yeah, thank you so much. So today I'm just going to talk about the work I did in Bogdan Pasternuk's lab at UCLA, along with our collaborators, where we applied this transcriptome-wide association study to 30 different traits to find novel risk genes. So, as we know, the most widely used technique to study genetics of complex traits, the genome-wide association study, where genetic variants are measured and directly tested for association with trait. So despite their success in identifying regions of the genome that correlate to disease status, GWAS often fails to provide mechanistic insight into what's driving risk for these traits. So to bridge this gap, we can rely on additional sources of information, such as QTL studies that connect genetic variation to molecular phenotypes, such as gene expression. A natural question we can then pose is whether we can correlate gene expression to disease risk directly, as these are more interpretable and testable biological units. So here we have our genotype data. For a subset of this cohort, we have gene expression measurements. So we can perform UCTL studies to associate SNP to gene expression, or we can take gene expression directly and associate this with disease status. Now, there are obvious technical challenges, not only in identifying the relevant tissue for the trait we're investigating, but also collecting it in sample sizes large enough to have sufficient power to detect an association. So how can we bridge this gap? Well, one thing we can do is just impute or predict the missing gene expression for the rest of the individuals using models trained on genetic data and external expression panels. And once we've done that, we can go ahead and correlate the imputed gene expression levels to a phenotype of interest. And in addition to just being able to predict into large cohorts, there's also the benefit of using the predicted expression and associated disease status, because we isolate the genetic component of gene expression. So that reduces the total variance of the trait of interest, and therefore we should have more power to detect association. So why is this important? Well, it establishes also a plausible chain of causality that directly links genetic variation to gene expression to trait under certain assumptions. So there have been a number of studies in the past few years that have been looking into this sort of approach, and they're all different ways to sort of view this sort of framework through different lenses. So there are at least a couple of potentially useful interpretations that I'm just going to just briefly mention. So first, Zhu et al. Pointed out this can be viewed as a two-sample Mendelian randomization test, which uses the top EQTL SNP as an instrumental variable for gene expression, which mediates the effect on trait. Alternatively, Gusev et al. Is my cursor working? Yeah, there we go. Gusev et al. Utilized the entire locus under a multivariate model, and this can be viewed as a test for genetic correlation between expression and trait if you're using predicted expression. For imputation-based methods, the statistical engines that predict gene expression can handle sparse architectures of cis gene regulation by incorporating different priors on effect sizes as well as other functional features. But maybe the most important thing that we can think of is that because these prediction models are linear, we can work with summary data directly without any real loss in accuracy, which means that we can leverage extremely large GWAS sample sizes that are currently available by just operating over the summary statistics. So with this in mind, for the remainder of the talk, I'll focus on our investigation for into 30 large-scale GWASs. So we studied a diverse collection of phenotypes such as height, BMI, schizophrenia, various lipid levels. So here in this table, we just have a couple sample traits, the sample sizes from each of those GWASs, and the number of risk loci that we found in those traits using 1 megabase as windows. So we compiled a compendium of data that covered more than 1 million individuals in total with 2.3 million phenotypic measurements. So overall, these GWASs had about 1,500 significant loci. So that's one element of the equation. And in addition to the GWAS data, we gathered 45 different gene expression panels, sampled from over 4,000 individuals. These were taken from various studies that all probed different tissues. So for example, we had RNA-Seq data from the GTEC consortium. We used 41 tissues from that study. The sample size for each of the expression panels was roughly about 150. We also utilized microarray data in blood, and together we integrated collectively we integrated these expression panels with the summary GWAS data and performed 30 transcriptome-wide association studies. Okay, so what did we find? In total, we identified 1,196 unique target genes across all traits and tissues. So in height, for example, we found over 700 target genes alone. Of course, this isn't completely surprising given our understanding of how polygenic height is, but we still find it interesting nonetheless. Also, there were approximately 400 genes that impacted multiple traits, suggesting a shared genetic basis at the level of gene expression. So in total, we had close to 1,800 associations. Some of these were repeated across multiple tissues, which provide evidence that there's some sort of shared effect across cis expression. So now that we've identified these risk genes, what can we actually do with this? Well, one example is fine mapping GWAS risk loci. So here I have a cartoon of the GWAS association peaks at 3 genes, A, B, and C. And in addition, I've drawn out the corresponding EQTL measurements for these genes in some tissue of interest. So to be clear, the y-axis on top represents the strength of association between genotype and phenotype, while on the bottom, the y-axis represents the strength of association between genotype and expression levels for these 3 genes. So just by looking at this toy cartoon, there's no clear way to discern which gene is important for the trait just by kind of seeing what falls within a risk loci. As there's 3 genes that have EQTLs, there's just by looking at the overlap, there's no sort of systematic approach that we can take. However, if we incorporate LD, TIWAS gives us a principled way to fine map genes which are impacting traits. So here we see that it's actually gene C that's driving the risk, despite the fact that offhand there seems to be a large overlap between gene A and risk for disease. So with that in mind, our data has approximately 1,500 GWAS hits and around 90% harbor cis EQTL. So again, this isn't surprising as emergent data from GTEC consortium and other studies suggest that every gene will have some sort of cis EQTL, sample sizes grow larger. The implication being that taking the gene at the GWAS locus that has an EQTL on its own is not an effective strategy for understanding the mechanism that's driving risk. Because TIWAS connects the cisgenetic component that impacts both gene expression and trait, we can prioritize target genes and refine a list of candidates to investigate in functional follow-up studies. So this is evidenced by the fact that only 36% of the 1,500 GWAS loci had at least one TIWAS target gene that met transcriptome-wide significance. So the target genes we identify are typically not the closest ones to any lead GWAS SNPs. So this suggests that the heuristic of gene assignment based on proximity may not be optimal. So in addition to be able to fine map known GWAS risk loci, we were also able to identify 168 novel risk loci that did not fall within 1/2 megabase of previously reported GWAS hits. So this was relatively robust to window size. We also looked at 2 megabase windows, and that number didn't change much. I think it went down to like 150. So the expectation of these findings is that these loci will be uncovered by larger GWAS studies, as TIWAS has more power. So what we did is we looked into a newer GWAS for education attainment that had over 400,000 individuals, and we were able to replicate all 7 loci we discovered using TIWAS in a data set with only 130,000 individuals. As another external validation, we examined the overlap between our results and an existing report in Genome Medicine that came out recently. So of the 12 traits that overlap between our study and theirs, we recapitulated 40 out of the possible 51 target genes they identified. So one thing I'd like to point out is that for these traits, we used, obviously an independent method and independent expression panels, which was pretty interesting on its own, right? In addition, we also uncovered more than 400 target genes. So this part is due to the fact that we had access to larger expression panels to run imputation into. Likely the reason for this large number of this large power gain is that TIWAS is a multivariate approach, and so it essentially can leverage allelic heterogeneity, or at least apparent allelic heterogeneity due to untyped causal variants. And so this is supported by the fact that we observed more than 200 genes where the gene level association is more significant than the top SNP association. So I just want to stress that this isn't direct evidence for allelic heterogeneity. Again, if a causal variant is untyped, it just could be that multiple SNPs in the region tag the underlying causal better. Okay, so with that said, we wanted to see if we can go beyond the set of target genes and ask whether TIWAS can provide any insight into the shared genetics between pairs of traits. So take, for example, in this illustration, we have two traits. The purple dots are the SNP effects on traits, and the green boxes represent the gene level effects on traits. So we can interpret the TIWAS association statistics as simply a causal effect of gene expression on trait, again, under certain assumptions. So to get a measurement of the genetic correlation at the SNP effects between two traits, roughly we can think of just correlating the SNP z-scores, after taking LD into account to get some measurement. We can also correlate the gene effects across traits to get a genetic correlation across expression. Okay, so we went and did that for all pairs of traits in our study, and we obtained genetic correlation at the level of SNP effects using LD score regression, and then genetic correlation at the level of expression using a method we described in the paper. So here I plot the values for each point in this plot is a study, and it plots the predicted gene effects, genetic correlation versus those found via LD score. So using our approach, we found 43 pairs of traits that were significantly correlated at the level of expression. And not only that, we see a striking similarity between our results with those found directly through the SNP effects. So we verified that this was meaningful using multiple permutations across the underlying expression levels. So taken together, these results may suggest that at least a portion of the observed genetic correlation is mediated through gene expression. So finally, we're interested in asking whether we can establish a possible causal link between two traits using the predicted expression levels. So here we have two causal models. The model on the left illustrates a scenario where genes influence trait 1, which in turn have an effect on trait 2. And the model on the right is the same thing, but going in the opposite direction. So we'd like to be able to distinguish between these two possible models given the TIWAS effect sizes. Right, so the way this works is that if the model on the left holds, the causal gene set for trait 1 should have a non-zero effect on trait 2. Right, so in this plot, each point represents a causal gene for trait 1 with its effect size on trait 1 versus the effect size on trait 2. And we can just fit a regression line. And we would expect to see a non-zero slope, given that we know this direction holds. If we then ascertain on the causal genes for trait 2 and perform a regression, we should see something in line with the null. again, because trait 2 does not directly impact trait 1. So we obviously don't have the causal gene sets in hand, but we can just use the target genes found through TIWAS as proxy and run a bidirectional regression using these gene sets. So as a sanity check, we confirm that triglycerides are causal for total cholesterol, which is to be expected, considering that total cholesterol is a direct function of triglyceride levels along with LDL and HDL. In addition, we're able to confirm a previously established causal link going from BMI to triglycerides and find additional plausible links going from triglycerides to LDL. So again, I want to stress that while these analyses are consistent with putative causal models, we cannot rule out additional models such as those where the genetic correlations are driven through shared pathways or pleiotropic effects. So in conclusion, we use integrative analysis to identify almost 1,200 target genes for 30 complex traits. We use these target genes to both refine known risk loci and also discover new ones. We then use the predicted gene effects to contrast pairs of traits and infer biologically plausible causal relationships. So I encourage you all to check out the preprint if you haven't. This work's currently under review at American Journal. We're still waiting on their decision. Thank you all for your attention.
Thank you, Nicholas. This was very, very interesting. Are there any questions? Okay, I assume there everything has been perfectly clear. What I didn't understand, maybe I asked a question. So what about the power compared to like GWAS, classical GWAS studies? So basically you have this effect for the school education or entertainment. Okay, you already showed it.
Yeah, I may have gone through that a little quickly. So here, for all the 30 traits, we have the number of identified GWAS loci. This indicates how many of those loci had at least one E gene, so just gene with heritable expression levels. And then so of these risk loci, 500, approximately 550 had at least one TIWAS gene. But in addition, overall, we identified approximately 1,200 risk loci, of which 168 were novel. So I think that our approach is sufficiently powered. We can find new risk loci, and then of these loci, obviously we wanted to sort of validate these, and the way we went about that is we went into a newer, larger GWAS cohort and said, okay, of these new loci that we found in education attainment, they should be found in a larger GWAS cohort, independent of expression data. And that was the case when we looked at that.
That's very interesting. Do you know if these genes have an overlap with those which have been found like in inter-disability studies?
I'm sorry, I didn't hear that. Could you repeat that?
And the genes which, or the loci, the gene loci which come up in the education entertainment study, are these correlated with those genes which come up in the severe inter-disability studies?
Oh, I don't think we looked at that yet.
Okay.
But that'd be an interesting analysis to perform.
May I ask a question, Dennis?
Yes,
sure.
Very basic question, but two questions. What is your comment about sample sizes? How far does such an approach reduce the how well does it increase the power? Yeah. In compared to a GWAS study. The other question is what I have not figured out in my brain yet is how come it does work if you are changing organ systems? So what if you don't have any brain samples or expression samples directly from the brain if you are investigating a brain disorder. Could you explain a little on that?
Yeah, sure. Those are both awesome, great questions. So for the first point about the power issue is that when we, using this TIWAS approach, right, I'll use educational attendant again as an example. The GWAS study that we used within the TIWAS framework had about 130,000 samples. You know, under TIWAS, as we kind of aggregate the signal across loci using these weights learned through expression, we had approximate power of 400,000 sample GWAS cohort, right? So that's one thing. Another thing also that gives you more power is also the threshold for significance is lower, right? As we're testing, you know, maybe tens of thousands of genes as opposed to millions of SNPs. So the Bonferroni correction is much less stringent at that level. So to answer your question also about the using the kind of relevant tissue for your study. So one thing that we also looked into, actually I have a slide, is how consistent is the effect size when we looked across tissues, right? And so one of the things that we looked at then was say, okay, fine. For each of the TIWAS results across tissues, how correlated were there? And we saw a very high correlation on average. And we think that just this is consistent with that the cis effect on expression and then therefore its eventual impact on trait is highly consistent. And this is kind of in line with the recent GTEC studies showing that, again, the cis effect seems to be shared, and what sort of differentiates tissue is going to be the trans effect, right? And since the TIWAS only utilizes the cis component, we're not going to be able to predict or sort of capture that tissue specificity until we get maybe to much higher sample sizes.
But to me, that is unexpected. Maybe I have an understanding problem there, but that this is only a cis effect, is that plausible? I mean, I know you proved it and everything, but how can that be understood on a biological term?
Yeah, I think at least in a biological level, we can think of maybe just the fact that I guess at the cis level, the level of regulation that these genes go through may be better mediated by the trans effect. So I'm not sure, maybe at least how to answer that in terms of biologically. That's something that we should probably look more into. But in terms of our results, you know, we did see a very consistent signal across all the tissues, which is in line with other studies.
Oh, great. Just for me personally, it's unexpected, but I mean, good say that.
I think it's unexpected for everyone. At least we were surprised. We definitely weren't expecting to see correlation that high.
Okay, thank you.
Okay. Yeah, thank you, Nicholas. And also thank you, Roland, for taking over the moderation. Yeah, we have recorded this. I will put it likely online the next days. And yeah, I wish everyone a nice weekend. And I think this was the last session. I think next week is also another one. Okay. So anyways, I wish everyone a nice weekend, and bye-bye.
Thank you.